Hi everyone, welcome back. A lot of VCE biology students miss out on easy marks in short answer questions simply because of their word choices. For example, if you say Caspases dissolve or kill the cell, that is a classic 0-mark answer in the eyes of VCAA examiners. In today's video, we are breaking down the cell biology and apoptosis questions from the 2018 NHT exam. I'll show you the exact scoring formulas and precise verbs you need to secure full marks under tight timelines. Let's get straight into it. Organelle identification. The image depicts the Golgi apparatus, or Golgi body. Key visual identifiers. Flattened cisternae, a series of stacked, curved, fluid-filled membrane sacs. Budding vesicles, spherical transport vesicles pinching off from the edges of the cisternae seen clearly at the bottom, which transport the processed molecules. The primary physiological role of the Golgi apparatus is to receive proteins from the rough endoplasmic reticulum, modify them, such as via glycosylation, adding carbohydrate chains, and package them into secretory vesicles for exocytosis, or transport to other cellular destinations. The correct answer is B. Modify and package proteins into secretory vesicles. Why the alternatives are incorrect? A is incorrect. This describes the mitochondrion, the site of aerobic cellular respiration, where the majority of ATP is generated. C is incorrect. This describes the network-like function of the endoplasmic reticulum, ER, or the general intracellular transport system, rather than the packaging hub of the Golgi. D is incorrect. This describes the function of ribosomes, which translate mRNA and assemble amino acids into polypeptide chains. The question asks which molecules can cross the plasma membrane by passing between the phospholipid molecules. This specifically describes the process of simple diffusion. The correct answer is A. Carbon dioxide molecules. Why? Nature of the bilayer. The core of the plasma membrane is composed of the hydrophobic, fatty acid tails of the phospholipids. This creates a significant barrier to most water-soluble substances. Characteristics of carbon dioxide is a small, non-polar, lipophilic molecule. Because it is uncharged and small, it can easily dissolve in the lipid bilayer and slip between the phospholipid molecules down its concentration gradient without any assistance. Why the alternatives are incorrect? B. Molecules of an enzyme. Enzymes are large, complex proteins, macromolecules. They are physically too massive to fit between individual phospholipids and typically require bulk transport mechanisms, such as exocytosis or endocytosis. C. Hydrophilic molecules. Hydrophilic, water-loving, or charged molecules, such as glucose, amino acids, or ions, are strongly repelled by the hydrophobic core of the membrane. They require transmembrane transport proteins to cross via facilitated diffusion or active transport. D. Molecules of mRNA. mRNA is a large, highly charged nucleic acid molecule. It is synthesized in the nucleus and moves into the cytosol through specialized nuclear pores, rather than crossing the phospholipid bilayer directly. A cell can be stimulated to undergo apoptosis after receiving an external signal that binds to a specific receptor on the plasma membrane. Below is a simplified diagram that shows how an external signal for the death receptor pathway begins to stimulate apoptosis. Caspases are a family of protease enzymes, specifically cysteine-dependent aspartate-directed proteases. They are synthesized as inactive precursors called procaspases. Once activated via a signaling cascade, such as the extrinsic death receptor pathway shown in the diagram, they function like molecular scissors. Pedagogical logic ladder, syllogism for students. Premise 1. Enzyme identity caspases belong to a class of enzymes called proteases. Premise 2. Biochemical action. Proteases function specifically by breaking down, or cutting, cleaving proteins. Conclusion. Application to target. Therefore, the direct action of caspases is to cleave or digest specific intracellular proteins, such as the cytoskeleton, or nuclear proteins, to dismantle the cell. High-value keywords to encourage. To ensure the mark is captured cleanly under tight assessment timelines, students should use precise verbs, cleave or digest, or break down, with respect to proteins. Specifying the location or targets, intracellular proteins, cytoskeleton, or nuclear structural proteins. Common student misconceptions to correct. Crossmark the melt-dissolve trap. Weak responses often state that caspases dissolve the cell or kill the cell. These are too vague for VCE standards. Students must identify the molecular target, proteins. Crossmark confusing caspases with phagocytes. Students occasionally write that caspases engulf or eat the cell. It is critical to reinforce that caspases execute intracellular digestion, whereas external white blood cells, like macrophages, handle the intercellular engulfment of the resulting apoptotic bodies via phagocytosis. Once caspases have carried out their action, the plasma membrane of the apoptotic cell forms apoptotic bodies. Phagocytes are attracted to the area, as shown below. Explain how the phagocytes shown in the diagram above perform their role. This is a 2-mark question that requires students to explain a sequential process: how a phagocyte removes cellular debris. Phagocytes, such as macrophages or neutrophils, are part of the innate immune system. In the context of apoptosis, their role is to clean up the environment so that cellular contents do not leak and trigger inflammation. The exam scoring formula. Because this is an explain how question worth 2 marks. The rubric expects a clear 2-step chronological chain. Mark 1 equals engulfing the apoptotic bodies via endocytosis or phagocytosis. Engulfment, Mark 1. The phagocyte recognizes eat-me signals, like phosphatidylserine, on the surface of the apoptotic bodies. It extends its plasma membrane around them, engulfing them via phagocytosis, a specific form of endocytosis. Mark 2 equals digesting, or destroying them using lysosomal enzymes. Digestion, Mark 2. Once inside, the vesicle, phagosome, fuses with a lysosome containing hydrolytic enzymes. These enzymes break down and safely recycle the organic molecules. Ideal student response, full marks. The phagocytes perform their role by engulfing the apoptotic bodies via endocytosis. Once inside the cell, these bodies are fused with lysosomes and digested by enzymes. Common student pitfalls to avoid. Crossmark missing the mechanism. The phagocytes eat the apoptotic bodies and they disappear. Too vague. Needs the term endocytosis for phagocytosis or engulf for Mark 1. Crossmark stopping halfway. The phagocyte surrounds the apoptotic body using its membrane and takes it inside. This only earns 1 mark. It fails to state that the cell digests or destroys the body once inside. Core concepts recap. Golgi apparatus. Q1 modifies and packages proteins into secretory vesicles. Simple diffusion. Q too small, non-polar molecules such as carbon dioxide slip between the phospholipids. Apoptosis, Q4 programmed, tidy cell death triggered via the death receptor pathway. Caspases, Q4A protease enzymes that cleave intracellular proteins, e.g. the cytoskeleton, to dismantle the cell. Phagocytes, Q4B engulf apoptotic bodies via endocytosis, then digest them with lysosomal enzymes. Exam tips and common pitfalls. Use precise verbs, cleave, digest, engulf via endocytosis, not vague terms. Name the molecular target, proteins, cytoskeleton, not just kills the cell. For multi-mark, explain how answers, give a clear step-by-step chain. Crossmark, dissolve or melt the cell, too vague for caspase action. Crossmark, confusing caspases, intracellular digestion, with phagocytes or engulfment. Crossmark, stopping at engulfment, without stating digestion by lysosomes. If you need my help, please add me on WeChat or WhatsApp, and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.